Hello biology champ, I warmly welcome you on another video at Chlorophyll classes where we are doing what? We are trying to maximize your centum probabilities by choosing and by revising and practicing through one of the best hot conceptual questions from the chapter human reproduction for today. This is a 13 videos, 13 chapter playlist which is going to come up for your biology board examination on this channel. Well, let's just start with our today's all the questions one by one. Well, the first thing that we're going to be studying in this is that we have segregated the whole into different topics. The first topic is the male reproductive system. And if you have prepared it, revised it, now let's test it that how better you are ready for your board examination. Let's go with the first question. The first question is that the testes in humans are located outside the abdominal cavity in the scrotal sac. Yes, that is true. Why it is present? So that it can help in the spermatogenesis process, because sperm formation can take place 2 to 2.5 degree less than the body temperature. So let's see that what is the first question on the basis of the given text, that is why this is extra abdominal position essential for the sperm production. So how you going to be writing it, the answer? The first answer that going to be going up will be like this. The first one would be that as for spermatogenesis 2 to 2.5 degrees Celsius less temperature is required than body temperature. Then it is saying that what is the optimal temperature? So optimal temperature is this. So you will write it. Next point, optimal temperature will be 2 to 2.5 degrees Celsius lesser. And if it is 2 to 2.5 degrees Celsius lesser, which basically means what? It means that around 34 to 34.5 degrees Celsius. Okay, moving on to the next one. The next second question of this, that is a man undergo a operation where one testis is surgically pushed inside the abdomen. So it has what, you know that this is, if it's a, this is a scrotum and these are the two testes present. So doctor did what? Doctor pushed one of this into what? Into the abdomen. Okay, so I am saying that this has been pushed into the abdominal area. Now what will happen? In this testes, the temperature that is required will not be fulfilled. So what will happen? So predict the long-term consequences of this on the fertility and the type of cells mostly affected. So the type of cell that will be affected, that will be spermatogonia, as they will not be able to change or develop into the sperms. Okay, so these are the affected cells. And when it comes to a long-term effects would be what? That since the with age we know that what happened, the sperm formation role will be reduced. Okay, now since if the two testes are there, it will be compensated, because you know that what is needed, a particular amount, 20 million sperm should be there. Okay, if it is less than 20 millions, then what we used to say, that we used to say that the person is infertile. So that infertility chances will come up much earlier. Okay, then the next question is crypto-orchidism. Crypto word is from the cryptocurrency, and which does, what does that mean? Crypto means hidden, orchid means is for the testes. So this is the conditions in which the testes are hidden. Where it is hidden? Testes are hidden inside and closer to the kidney part, because they fail to descend into the scrotal sac. Okay, so why is this cause of the male infertility? The major reason for this to cause infertility would be what? That the temperature is not being suitably present for the, because the temperature would be what? 37 degrees Celsius in the testes, whereas the temperature required would be what? 34.5 degree. Okay, close to that. What type of cell would be still functional, relatively normal? Your leading cells will be functional. Okay, leading cells will be there, interstitial cells will be there. These cells would be comfortably working, means testosterone will be produced. Only the sperm formation won't be there. Okay. Moving on to the next one. This question says that the seminiferous tubule are the site of the sperm production and are lined by two types of cells. What are the two types of cells? One is your leading cells, another one is interstitial cells. Okay. Now name the two types of cells lining the seminiferous and state the function of each. Oh, wait. Seminiferous tubules are the site of sperm production and lined by two types of cells. Means that inside it there are two types of cells. Okay. So number one, there is what? Spermatogonia, which will be what? This will be precursor of sperm cell. Or it is what? Sperm mother cell. Whatever you wanted to call it. Then the second type of cell that is present is the Sertoli cells. What is the role of the Sertoli cells? Sertoli cells do the job of what we say, nourishment provider to growing cell, or it also forms at what? Blood testis barrier. It secrete androgen factors which inhibits the growth of Mullerian ducts also in the males, and many more such functions. Okay, moving on to the next question, that is the interstitial cells lie between the tubules. Okay, what do they secrete? Interstitial. Okay, second is leading cells or interstitial cells. They secretes what? They secretes androgen, testosterone. Basically concentrate the testosterone levels. And what is the role of its secretion? It helps in the sperm formation. Okay, or secondary sexual character development. Okay, next is a patient has a normal leading cells function. Okay, but damaged Sertoli cells. Predict the effects on testosterone level. Now you know that leading cells are responsible for these testosterone formation. So testosterone will keep on forming and its level will be super high. What will be the sperm production? Since Sertoli cells are not there, they are not able to provide the basic nourishment. Without nourishment, there won't be any growth. So there will be no sperm formation. Okay, this would be no. This would be normal. Comes to the secondary sexual characters that will be formed because that is linked to what? Testosterone levels, which are normally being formed. So it will develop. It has nothing to do with the sperm formation. Okay, moving on to the next one. Trace the path of the sperm formation for the site from the site of formation to the point of ejaculation, naming each duct in sequence. For each structure, state one function it performs in the sperm maturation and transit. So they have given the track also over there, and then you have to tell it. So first one, seminiferous tubule. There is what? Formation of sperms. So here the sperm formation take place. Then reti testes. These are what? These are the network of ducts from seminiferous tubule. They do what? Transient the sperm from this to next. This from this to next means the seminiferous tubules to the vasa efferentia. Same vasa efferentia. These are also what? Networks of ducts from reti testes to outside into the epididymis. Then comes the epididymis. What is the role of epididymis? It's stored and mature. Store and mature the sperms. Okay, moving on, then vas deferens. What is the role of this? Transport from testes to outside into the accessory duct. Sperms transport. Okay, next is your ejaculatory duct. It has what? Secretion of seminal vesicle plus the vas deferens. So it is made up of this. So it contains what? It contains the enzymes, it contains the secretions of the seminal fluid and forms the 60% part of the seminal fluid and do the things. Then comes the urethra. Urethra is nothing but the passage to pass through it. External urethral orifice. What it is? It is through which it goes outside the male body. Sperm goes outside the male body. Okay. Also, why is epididymis crucial for the sperm maturation even though sperms are formed in the testes? Because in the epididymis, proper tail formation, we used to say that used to occur over here. Okay. Without tail, it won't be completed. Going to the next question. Study the following clinical scenario and answer. Patient A has a normal testosterone, no sperm, like testosterone is normal but no sperm is present in the semen. This condition is called as azoospermia. Epididymis normal, vas deferens is blocked bilaterally. Means most probably what happened, that there will be sperm formation normally, but outside the sperm are not coming up because of the blockage thing. Okay, so and you have to identify the most likely the site of problem for each patient. Here the site of problem is the blocked vas deferens. So first is vas deferens. Second case, low testosterone, normal sperm count, Leydig cells non-function. So what is the problematic area? Seminal tubules, seminiferous tubules. Then in the third case, patient C, normal testosterone, sperm count is low, Sertoli cells shows inflammation, means there is a problem with the Sertoli cells. So how do you gonna be tracking this thing up? You're gonna be tracking this thing up with what? That there is problem in the Sertoli cells. Okay, so we have identified the site where the problem is. Sertoli cells are present in what? Seminiferous tubules. Now the second question say that for patient A, can sperm be retrieved from IVF, from which structure and why? Since in the first case sperms are being formed, later on they wanted to have a baby, what we can do is that we can directly take out the sperm from the testes where it is being formed. Okay, so what gonna be happening in the second answer? What you're gonna be saying that yes, we can retrieve. Please students, always remember that whenever you are giving the answers, try to give it in a as many, like what we say that as written form as possible, but underline the important parts. Okay, so just underline that yes, okay, we can retrieve sperms from which structure? Testes. Okay, mark this up, not with a circle but with a underline, from the testes. And why? Because sperm can be used for the in vitro fertilization technique. Okay, then comes the third question. Its answer is, for patient B, how can testosterone levels be normal if the Leydig cells are non-functional? Suggest what might be the compensating point. Okay, so here what happened, that adrenal gland also secretes, adrenal glands also secrete androgenic hormones which will help in this formation. Okay, then comes the fourth question, that is, for patient C, why would Sertoli cell inflammation reduce the sperm count? Because of the, as it is responsible for the nourishment. Okay, name two specific function of the Sertoli cells that would be disrupted. So two functions I have told you the last time itself. One is the nourishment, second is what, blood testes barrier is there, or other functions are there that will be problematic in their cases. Okay, coming on to the next question. The accessory gland of the male reproductive system consists of what? Your seminal vesicle, prostate gland, bulbourethral gland collectively produce seminal plasma. Okay, we understood this thing. Now, what percentage of the semen volume is contributed by the seminal vesicle? 60% is contributed by the seminal vesicle. What does their secretions contain and why it is important from the sperm survival? You know these questions. Okay, these are straightforward. What they contains? They contains the fructose, they contains the prostaglandins, all of those things which are present on it, you have to write it down. Okay, then comes the prostate gland secretes a slightly alkaline fluid. Why is the alkaline important for the sperm in the female reproductive tract? So the reason is female tract is acidic. So to neutralize it, alkaline medium is supplied so that sperm can survive. Coming to the next one. Next question is, bulbourethral glands, also called as Cooper's glands, secretes a lubricating fluids before ejaculation. Why is this important? What would happen without this secretion? So basically what happened, this lubricant secretion lubricates penis and it lubricates the penis and the urethral orifice so that it reduces the friction while ejaculation. Okay, to reduce friction. Then the next one is, a man has had his seminal vesicle surgically removed. Okay, predict the effects on the semen volume. If the seminal vesicle is removed, semen volume will decrease about what? 60% decrease, because the 60% is coming from what? Seminal vesicle. Then what about the sperm motility? With respect to sperm motility, that will also reduce because the calciums that would be there won't be coming up. Then fertilization success also reduced because of these things which are not present to it. Moving on to the next question. Critically analyze the structures and the function of the male reproductive systems by the answers. Now these are the similar questions that we have done. Let's just go towards the lower side or the last question. Okay, this question we have not discussed, here we can discuss it. That is, FHS and LH are released from the anterior pituitary. Explain the specific target cells action feedback loop for each hormone in the male reproductive system. How you gonna be writing it up? You're gonna be making, not writing the paragraph or the pointers. Of course that you have to do it, writing the pointers. But first make a 1 page or the half page flow chart of it. So basically what happened? You have your hypothalamus. Hypothalamus will release gonadotropic releasing hormone, which will act on the anterior lobe of pituitary. Now this will release two hormones, FHS and the LH. The FSH will act on the Sertoli cells. And the LH will act on the Leydig cells. This Leydig cell will secrete androgen, that is testosterone. Okay, And this testosterone as well as Sertoli cells together will do what? Spermatogenesis, means sperm formation. Now what happened? When the testosterone level increases, it gives what? It gives a negative feedback to both of this. Okay, so who will get the negative feedback? These two, like the high level of increase level of testosterone will give what? A negative feedback. But if it is reduced, it will give what? A negative, it gives a positive feedback. Along with that, it also secretes a hormone called as inhibin. So if the spermatogenesis process is like that, too much sperms are being formed up, this inhibin also go and do the same negative feedback. Okay, but by the time the sperm count goes down, it gives what? It gives a positive feedback. Okay, so that depends upon the situation. So you have to make this and write about it. Okay, moving up to the next topic, that is a female reproductive system. Okay, so in this we have 4 questions, basic questions are there. Let's try to do it. The first one is from the uterus. It's saying that uterus is what? A pear shaped organ that houses the developing fetus. Analyze the following. Number 1 is that the uterine wall has 3 layers. Name them from outer to the inner and state its function. So these kind of questions, you can even make the diagram to support your answer much better. Okay, so this is your what? Female reproductive tract. So I'm going to make it, this layer is called as what? The outermost layer. So we have to write the outermost, middle and the innermost layer. So outermost layer is called as what? Perimetrium or epimetrium. Then we have what? A thick muscular layer. This layer is called as what? Myometrium. Myometrium. And the last one layer, innermost layer, is known as endometrium. So you're going to be writing it up as the endometrium. And state its function. Perimetrium do what? It forms the protective lining, protective outer lining of the organ. Okay. Then comes to the myometrium. These are what? Muscles, smooth muscles are present, help in the contractions and the baby coming down. Endometrium is what? It provides a site for implantation. This is the function of it. Next one is the endometrium undergoes cyclic changes. What triggers its proliferation and what triggers its shedding? So its proliferation is triggered by the progesterone and the estrogen. Okay, primarily the estrogen. The second one is proliferation by the estrogen. And who is giving this estrogen? Follicles. Okay. And what triggers its shedding? Shedding is being triggered by the unavailability of estrogen and progesterone. Okay. Moving on to the third. A woman endometrium fails to proliferate due to estrogen deficiency. Okay. What consequences would this have for implantation? They can't implant if there is no proliferation, no endometrium present. Implantation can't occur. What would happen to the menstrual cycle? So if there is no proliferation, means there won't be any menses or there won't be any bleeding that will take place because there is no endometrium present itself. Okay, I hope this thing is understood to you. Next, The fallopian tubes plays a crucial role beyond mere transport of ovum. Okay, what else it do fallopian tubes? That we say that it's just not do the transport of ovum. It is also the site of fertilization and the embryo development, right? Then what is the site of fertilization in human? That, as the question came itself, that is ampullary isthmus junction. Ampullary isthmus junction. Okay. Why must fertilization occur here rather than directly in the uterus? Because what will happen? There is a proper time needed when there will be the thing that blastocyst has to be formed, which takes 7 days to form. If we kept it in the uterus initially itself, that uterus cavity might do some contraction, it just goes down. Okay, there could be a contraction that could have happened because of the oxytocin levels if has been released in the body. So what will happen? We don't want that the developing embryo is not implanted, because until the 7 days over and the blastocyst kind of thing has been formed, it will be there. So what will happen? We don't want that. So that's why 7 days time is being required. Okay, so if someone will ask that why it is not directly implanted, so the reason is simple, that is only blastocyst has, because it has trophoblast cells which are sticky cells that can only go and join with it. Okay, because only blastocysts can implant in uterus, and it takes 7 to 8 days to form. Okay, until then any contractions in uterus might exempt early embryo outside the uterus. So implantation will not occur. This would be more like what? Miscarriage. Okay, the initial days miscarriage can happen. Then comes what? In the ectopic pregnancy, what is we did the second question. We have to, the fallopian tube is lined by ciliated epithelium and has muscular wall. How does these two features contribute to the transport of ovum and the embryo? So basically ciliated propel it towards that side. Cilia is propelling the things towards the uterus side, as well as the contractions are like a perisylastic contractions, right? So when the food is going down, same way that also contracts and push the things towards the uterus side. I hope this thing is understood. Then the third one, in ectopic pregnancy. What is ectopic pregnancy? Basically, beta, what happened that sometimes there are cases that the fallopian tube get your implanted. Okay, so that is the major problem. And then we know that fallopian tubes, if the pregnancy implantation has occurred in the fallopian tube, we call it ectopic pregnancies. So explain why this is a medical emergency and what structural feature of the tubes makes more implantation impossible there. First of all, complication is that since there won't be any blood supply which is needed, so and the space is also not there, their contractibility is also not there, that they can extend like the uterus. Okay, so what will happen if something is implanted over there? It leads to what? It leads to the busting of the fallopian tube eventually, and as it will keep on growing, it will burst the fallopian tube because of its small space, right? And that will leads to what? The death of the baby as well as the mother also. Okay, so this is the major problem. And what's the structural features of the tube that inhibit it? First of all, that presence of what? Your ciliated epithelium, which will propel it and don't let to be sink it or implant over there. Okay, moving to the next question, that is, the ovary has dual role as a gonad and an endocrine gland because it's releasing the hormones. Name the two hormones produced by ovary and state when in the menstrual cycle each is predominantly secreted. So the two hormones are what? estrogen and progesterone. Estrogen and progesterone. Okay, so the progesterone is secreted during the proliferative phase and secretory phase, whereas your progesterone is being secreted during the time of secretory phase. Okay, next, first one is done. Then the next question is, a secretion of the ovary shows follicles are different development stage. Okay, so we are seeing what? We are seeing the ovary, and we saw that there are different follicle in different stages. Name the stages from the primordial to the Graafian follicle in sequence. So we have what? We have primordial follicle. Then we have primary follicle. Make it like a flow chart. Okay, primary follicle. Then we have secondary follicle. Then we have tertiary follicle. Then we have what? Your Graafian follicle. Moving on to the next one. The corpus luteum forms after ovulation. What hormones triggers corpus luteum formation? So the answer to this one is the LH. Okay, triggers the corpus luteum formation. What does it secretes? And what is the fate of the corpus luteum if the fertilizations do not occur? Corpus luteum secretes progesterone and estrogen, which maintains uterine lining. So what will happen if it is degenerated? Uterus lining will be shed off. So fate would be what? Uterus lining shed off. Moving on to the next one. A woman has a polycystic ovary syndrome, which means that multiple follicles forms but none of them reaches to the Graafian follicle stage and ovulation never occurs. Predict the consequences on the estrogen level, progesterone level, LH and the menstrual cycle regularity. First of all, estrogen level. Since the estrogen is secreted during the initial phase, everyone is able to form certain things but not the Graafian follicles. Means the estrogen levels will be there. Okay, so estrogen levels will be higher. Progesterone level won't be there because no Graafian follicle has been formed. So that would be what? That would be least or not at all present. LH surge will occur, but no one will respond to it. Okay, LS cell will occur. Okay, but nothing will respond to it. Menstrual cycle will not be regular. That's why. Okay, so there will not be bleeding also, shedding also. Shedding and bleeding would be there, but the timing won't be very fixed up. Okay, moving on to the next one. That is, analyze the structure and the function of female reproductive systems comprehensively. Compare the anatomical position and the histological structure of ovary with the testes. Identify two structural similarities and two differences. So if you see that here, what happened, they are basically taking on the basis of the development of the testes and the ovaries. So if you see that anatomically and the histologically, there are the, like if it comes to the histologically, then there are what? There are the mother cells, okay, like oogonia or the spermatogonia that is present, which will be changed into a haploid cells. So this is the one similarity, histological similarity. Plus, what would be the next thing? That would be what? That there will be differences. If we have to see that, then there won't be the number of the cells that will be forming up, okay, in the per cycle. That would be very different. Okay, with respect to anatomical position, the germinal layer that is being formed is similar. That is enough. Okay, then the vagina, cervix, uterus, fallopian tubes, ovaries each play a specific role in the reproduction. Describe the specific function of each structure in the journey from insemination to the implantation. This is straightforward question. Okay. Then the cervical mucus changes consistency during the menstrual cycle, thin and watery at ovulation, thick and the sticky at other times. Explain what hormones control each changes, biological significance of each consistency type. So basically is what during the ovulation it is thin and watery so that sperms can pass through it. Plus rest of the time it is thick so that even if the sperm will go, there is no point because there won't be any reproduction activity that will take place because ovulation has not occurred. Okay, the hormones is not in your syllabus, how that's working. Okay, I'm sorry. Coming up to the next question, the zona pellucida of the secondary oocyte has glycoproteins ZP1, ZP2, ZP3. What is the role of ZP3 specifically in the fertilization? So basically what is what? Polyspermy and the cortical reactions that is being taking place. Okay, so that is what the answer would be. This is also straightforward. I hope that if you would have studied this, but this is beyond the NCERT level, so you can leave it also this point. Okay. Then a woman's fallopian tube are surgically tied. So there is a tubectomy case. Explain at which point is the reproductive process that prevents pregnancy. It is the mating during the fertilization time itself. Okay, ampullary isthmus junction, they can't meet. Does it affect her menstrual cycle or the hormonal balance? No, it doesn't affect her menstrual cycle or the hormonal balance because there is no change with respect to those major structures, that is hypothalamus, anterior pituitary, follicles or the ovary and the uterus. Okay, moving on to the next one. That is the third topic, spermatogenesis, that is male sperm formation. Let's start with the first question. That is, spermatogenesis begins at puberty and continue throughout the adult life. Explain what triggers the onsets of spermatogenesis in puberty. That is the release of testosterone. Okay, that is the whole hormonal cascade effects which we have already discussed before. So you can write that whole thing. Okay, that graph. Then the second is trace the cell lineage, spermatogonia, primary spermatocyte, secondary spermatocyte, spermatid sperm. State the ploidy and the type of the division at each stage. So ploidy would be at spermatogonia, that is 2n, that is basically it will be what, 46 chromosome. Then it becomes the primary spermatocytes. So primary spermatocyte is nothing but what, a differentiated form of spermatogonia. Okay, so this is also 46. Then the secondary spermatocyte is what, it is a haploid structure, so it will be 23. Then spermatid will also be haploid, 23. Sperm will also be haploid, 23. Okay, now the true division that they are going on, this is over here is what, it is just mitosis and basically differentiation, nothing else. Okay, moving on, this is what, where the meiosis 1 occur, here when the meiosis 2 occur, and here is what, this is just the differentiation. Okay, moving on to the third question. How many functional sperms are produced from one primary spermatocyte? From one primary spermatocyte there is 4 functional sperms formed. Compare this with the number of functional egg produced by one primary oocyte, that is 1 only, 1 ovum. Explain the biological significance in the difference, because most of the sperms would be what, would be the naturally selected out. Okay, because of what, because of the condition they had to go through in the female fetus, sorry, in the female reproductive tract. Whereas egg, since if more than one egg will come and more than one, it requires the resources later on for the development of the fertilized embryo. Okay, so I hope this thing is understood, the significance of it. Okay, next is spermatogenesis is the transformation of spermatids into the mature spermatozoa. What is the difference between spermatozoa and spermiogenesis? What does each process, where does this process occur? Very easy question. Spermatogenesis is the whole process. What is the spermiogenesis? It is a process of formation of spermatozoa from spermatids. Okay, that is, where does this occur? This spermatogenesis is occurring into the seminiferous tubules, whereas this spermiogenesis is the process which is occurring between the ducts and the epididymis. Next is what? During spermiogenesis, the spermatids undergo dramatic structural changes. Describe the formation of following structures and state the function of the each: acrosome, middle layer, and the tail and the flagella. So basically what happened, that this is your spermatozoa, spermatid. Okay, now what will happen? Its Golgi body, it change its into a cap like structure, that is what? Acrosome. Whereas most of the cytoplasm will be left out. Okay, so the acrosome is formed by Golgi bodies. Middle layer will be what? All the mitochondria will go and attached at this area. That's why the cytoplasm is getting very less, because they are extending themselves with the same area. So mitochondria will align themselves into what? Into the middle piece area. And the tail and flagella is being formed later on and thin. At the certain end, there will not be any cytoplasm also covering present. So it will arrange into 9+2 arrangement of the tubulin protein with cytoplasm around it. Next is the sperm head contains highly condensed DNA wrapped around protamines, not histone, which is a compaction necessary for the sperm cell. Okay, this is beyond. Okay, no need to do this. Moving on to the next one, hormonal regulation of spermatogenesis involves a precise feedback thing, which I have already explained you in the last question. So you can come back and see that again and do this question, the chart which I have made it. Moving on, there is what? A researcher conducts the following experiment on the male mice. In first experiments, it removes the pituitary gland, spermatogenesis stop, because FH and LH won't be secreting. Then experiment 2 injects FHS alone into the pituitary removed mice. Partial spermatogenesis resumes. Complete won't take place because of what? Because LH, which will be secreting the testosterone, is still not here. Okay, because of no LH. Next is experiment 3, inject LH alone into pituitary removed, testosterone increase, but spermatogenesis does not reduce because FHS is not there. Because FHS is not there, then who gonna be providing the nourishment? No nourishment, no development. Not activated or present. Then the fourth one, inject FHS and LH together, full spermatogenesis resume. Now, what does experiment 1 tells us about the requirement of the pituitary hormone in the spermatogenesis? This is nothing, just your hormonal feedback whole diagram, and those questions you can easily do it. Okay, let's go to the next one. Critically compare spermatogenesis and the oogenesis in the human under the following heads. When does the process begin? Fetal life, birth or puberty in each sex, and when it ends? In males, it started in puberty, okay, and it's keep on going on throughout the life. In females, it starts at the fetal age and it goes till the adulthood, okay, every month one. Compare the cell lineage, ploidy at each stage, and the number of functional gametes produced per precursor cell. This is just the diagrammatical representation given in your NCRT. One diagram is given where both the things are being highlighted. So that thing is being mentioned here. Okay. Then in spermatogenesis, meiosis is completely fully before the sperm are released. In oogenesis, meiosis 2 is complete only after fertilization. Explain the biological reason for this differences. So what would be the reason? The first thing is what? In the female, it is energy. This process requires energy. And why to do that thing? That if even the fertilizations don't occur. plus the male contains the centriole which is required, or contains the, sorry, it's not the centriole point over here, I'm sorry, I take my word back. Male contains the factors which promote this thing. So until those factors would be present, which will be bring out, which gives the surety that the fertilization will occur, it completes its thing in the egg. Whereas in the sperm, they are being made that they will be produced in enormous amounts, so the energy would be required in that case. Okay, so they complete the whole process anyhow. The wastes would be there, so they do the extra step also. Spermatogenesis is a continuous process, produce millions of sperms daily. Oogenesis produces one egg per month. Explain the cellular and the regulatory mechanism behind the quantitative differences. I have told you this thing, and you also know that what it is there. Both process requires both process required Sertoli cells and the follicle cells respectively. Compare the role of these supporting cell in the male and the female. Okay, in male, Sertoli cells help in what? Nourishment and all those things, you know. Follicular cells helps in what? It helps in the estrogen release, and as the estrogen release, it helps in making the uterine lining for the future implantation if it occurs. Okay, then what function and the analogies are, and what is the unique to each thing? Basic questions, you can do it. Okay, if you don't, then let me know in the Telegram. I will give you the link so that you can do that. Next comes to the oogenesis and the follicular development based questions. The first question is, oogenesis is a discontinuous process that begins in the fetal ovary and is completed only after fertilization as an adult. At which stage of meiosis is the primary oocyte arrested at the birth? It is the prophase 1. How long it can be arrested and persisted? And in the prophase also it is diplotin. Okay, until how long it persists? Until puberty, which is usually 10 to 13 years. Next comes, what event changes the primary oocytes to the resume the meiosis once during the menstrual cycle? The FHS secretions from pituitary gland. It's made it. Okay, then at which stages does the secondary oocyte arrest? It is arrested as the metaphase 2. Then comes the third question, when and where the meiosis to completed in the secondary oocyte? When sperm comes, and where it is the ampullary isthmus junction. Ampullary isthmus junction. Next come, what triggers this completion? Anaphase promoting factors. It promotes it. Okay, moving up to the next one. Follicular development in the ovary passes through several stages during the menstrual cycle. Name the stage of the follicular development in the correct order from primordial to the Graafian follicle. Already we have done it. Okay, then the antrum is the fluid filled cavity that develops within the follicle. What is the antral fluid? It is water cells. Okay, what does it contains that is important for the oocytes? Okay, this is not your level, so you can just leave it. Only one follicle typically becomes the dominant Graafian follicle despite multiple follicle begins development. Explain the mechanism by which the other follicles undergo atresia. Not in your point. Okay, coming up to the next one. Compare the products of the oogenesis and spermatogenesis. From one primary oocyte, how many cells are produced? 1. Name each cell type, which ones are functional. So basically they are telling that there will be 2 polar bodies and 1 secondary oocyte that will be formed, not ovum. Okay, why is 3 polar body produced during oogenesis? What is their fate? Actually not 3, but basically just the 2 polar bodies are being formed. Yeah, yeah. if the first polar body further divide, that is why it is considered as the three polar body is being formed, because it also has what, half of the quantity of the genetic material. So this is what you know that na, like there is what, oogonium forms primary oocyte. This primary oocyte divides into two. One is the polar body, one is what, secondary oocyte. Later on this secondary oocyte also divide and forms what, it forms your ovum plus second polar body. So they are saying that this will further divide, this can divide. Okay, so this is what, three polar body are being formed. Now the question is what, why they are being formed? To maintain or balance the quantity of the ploidy. Okay, what is their fate? They will degenerate. Is it energetically wasteful? Yes, it is. Okay, but it is important for the ploidy level maintenance. The second oocyte at ovulation is not a mature egg. This one. Okay, explain. Sorry, this one. Secondary oocyte which came out during the fertilization time, it is not mature enough because it's still restricted into what, it is still restricted into the metaphase. Explain what structure is actually released at the ovulation. Secondary oocyte. It is secondary oocyte that is being released. At what stage of the meiosis? That is metaphase 2. And what it is surrounded by? It is surrounded by the, name the layers from outside to inside. So you have to write about the ovum structure. So it has what, corner, if this is your ovum. Okay, it has what, it has a layer, corna radiata first. Then we have what, perivitelline space. Okay, wait. You have a vitelline membrane. Then there is a space called as perivitelline space. Then there is a presence of corna radiata. Then there is presence, zona pellucida. Then there is a presence of a cellular layer called as corna radiata. So this you can simply write up. Okay, next question. A woman is given a fertility drug that stimulates the multiple follicles to mature simultaneously. What class of drug? Okay, this is... First one is not in your syllabus. Second question, let's see that. Multiple graphene follicles are now present, all ruptured at ovulation. If the multiple eggs are fertilized, what type of twins or higher order multiples would result? Genetically identical or different? So actually they were what, multiple ovum is there, all the ovums will have what, different genetic material, so they will be what, genetically dissimilar. Okay, so they won't be the identical one, they will be more called as what, fraternal twins. Okay, then after retrieval of the eggs for the IVF, the corpus luteum still forms from each rupture follicle. What happened to the progesterone level? It will spike up. What would this affect the endometrium? It will maintain the endometrium. Okay, if all the egg retrieves were secondary oocytes, at what stage would they have needs to be fertilized to allow the meiosis to complete? What if all the cells retrieved for secondary oocytes, at what stage would they need to be fertilized? When the ovum will be formed, means when the completion of this whole structure would be occur, means basically they are all arrested in what, metaphase. So we have to complete that. What is triggers for that? What is the trigger for meiosis to completion? That is anaphase promoting factors coming from the sperm. Next is what they have: comprehend the fetal life of the birth of the child. In a fetal ovary, oogonia divide mitotically to form primary oocyte. At which stage of the fetal development does this occur? From the 5th to 7th week of development. How many primary oocytes are present at the birth? Millions copy of it is present. Why does this number decline significantly by puberty? Because of the atresia over the period of a time. They degenerate. Trace the complete development. Primary oocytes, secondary oocytes, mature ovum. Include ploidy stage of mitotic arrest. Triggers for the resumption of the each step. Straightforward questions. Okay, you have to just write it what is happening at the each level. So if you know the flow chart, you can do it. That's there in my first initial videos also on this channel if you go down. Okay, the zona pellucida forms around the okay. Now these two are not at your level. Next is the compare the fate of the secondary oocyte in the two situation. So fertilization occur and fertilization do not. If the fertilization occur, it will change into what? Zygote, then ultimately go the cleavage divisions and form some morula, blastula and get implanted. But if the fertilizations do not occur, what will happen? The cilia of the fallopian tubes, they will propel it towards the uterus and finally it will be come out with the menstrual flow or can even degenerate also at that point itself. Okay. Include what happened to the oocyte, the polar body, the corpus luteum in each cases. They will also degenerate. Corpus luteum will stay for 14 days and then it also slowly regress into the corpus albicans and degenerate one. Okay, moving on to the next one, that is the menstrual cycle. Now the menstrual cycle in itself is a topic where a lot of pointers are coming up. If you know that, those questions would be done, and assume that this is one of the important topic, very important topic. Okay, coming up to the next first question. The menstrual cycle is approximately 28 days and involves coordinated hormonal and the structural changes. List the 4 phases of the menstrual cycle in order and give the approximately day range for each. Straightforward question. We have what? Menstrual phase, which lasts for 3 to 5 days. We have the follicular phase, okay, where the follicular development takes place. It lasted, or the polyphilitic phase, which lasts from day 5 or day 6 to day 14. Then day 13, then the menstrual phase, then the ovulatory phase, which will last from day 4, which will happen on the day 14th. Then from day 14 to day 28, 15 to day 28 would be the luteal phase or the secretory phase. Okay, coming up to the next one, write it proper column. This phase, this days, this phase, this days. Okay, which hormone is responsible for triggering the ovulation? That is LH and FHS. On which day the LH surge typically occur? That is 14th day. And what event does it causes? The Graafian follicle rupture. Then comes a woman's cycle is 35 days long. On which days does the ovulation occur? Now you should always remember the luteal phase is very constant of 14 days. So you can tell the day of ovulation day will be what? Total number of menstrual days minus 14, that is 35 minus 14, that is 21st day. There will be what? Ovulation that will occur. Moving on to the next question. The corpus luteum is the one of the most important endocrine structure of the menstrual cycle. How does the corpus luteum form? It is formed by what? The condensation of the ruptured Graafian follicles into the corpus luteum. Which hormones trigger its formation? It will be the LH. Okay. Then comes what? What hormones does the corpus luteum secretes? It secretes progesterone and estrogen. How does it maintains the endometrium lining? Because progesterone and estrogens will help in maintaining the consistency of the progesterone for the endometrium lining for the 14 days. Corpus luteum is pregnancy has a long life. However, if the fertilization does not occur. It remains active only for 10 to 12 days, maximum 14 days. Explain the molecular reason for its regression when the fertilization does not occur. What happens to the endometrium lining? So if the fertilization do not occur, then what will happen? That your uh, corpus luteum only work for 14 days. But once the fertilization occur, the HCG will be start releasing up. That HCG and the chorion will support the corpus luteum to keep on forming what? Keep on releasing the structure. So this thing is the major thing over here working. Coming up to the next one. Hormonal regulation of the menstrual cycle involve complex interactions between the hypothalamus, pituitary and the ovary. Explain the positive feedback mechanism by which the rising okay, this is a hormonal, straightforward hormonal question. So if you will do that the same way we have done it for the spermatogenesis or the male reproductive system, same worked over here. Okay, so do that thing and that will how you can give the answer to these questions. Okay, if you don't able to do it, let me know in the comment section and I will help it. Same goes with this question and same goes with this question. Take a screenshot and all the menstrual cycle and their points and all those things are being covered over here. Try to do it. If you're not able to do it, I will give you a personal in Telegram. Okay, and its link, the you will search chlorophyll classes, you will get it, or you can get it in the links also in the description. Coming up to the next, that is fertilization and implantation. Fertilization is a multi-step process which the several crucial barrier. What is the site of fertilization? Ampullary isthmus junction. Why must the sperm reach the site for quickly after the ovulation? Because your ovum survives for certain period of a time. Okay, that is 72 hours. After that, it will die. So degenerate. So that's why. The sperm must undergo capacitation before it fertilizes the egg. What is the capacitation? This is certain morphological changes in the sperm for it to become capable for the division. So for example, its plasma membrane should be removed, faster movements. All of these things are what the part of the capacitation. What is capacitation? You know that it's the morphological changes to make it capable of fertilizing the egg. Where does it occur? It occurs in the uterus and the fallopian tubes. What structure changes in the sperms does it undergo? Removal of that thing, whip splash movement, faster movements and all those things. Only one sperm fertilizes the eggs despite millions of sperms being ejaculated. Describe the Zona reaction and how does it present the polyspermy. Okay, this is the polyspermy thing which is beyond NCERT at your level. So we are not going through it. You can leave it. Okay, this is for the NEET level, not for the board examination. The acrosome reaction is a prerequisite for the sperm penetration of the egg. What is acrosome? It is a cap-like structure present on the head of the sperm. What triggers the acrosome reactions? So acrosomal reactions are what? Once it has to bypass the corner radiata, zona pellucida. So all these layers contains what? Cementing materials and all those things. So we have to remove it. Like hyaluronidase will be there, which will remove the cementing material between the corner radiata cells. So this thing will be the one thing. Then the next question is describe the sequence of events, acrosomal reactions, penetration of zona pellucida and plasma membrane fusion. In detail they are asking up, which is not there in our syllabus. You can leave it. Next question, a person's sperm has normal morphology, normal motility, but fails to penetrate the zona pellucida of the egg in the IVF. Which specific step is defective? Okay, this is have normal morphology, normal motility, but fails to penetrate the zona pellucida. Now, zona pellucida penetration is important step by what? By the zona pellucida reactions. Okay, so probably there would be what? Incompatible sperm could be there. Okay, that could be one of the reason. Okay, or there could be what? There is the acrosomes do not have the all the hormones or the enzymes, which is not the hormone but the enzymes. And how, which art technique we can use to bypass? We had to take another sperm. So from the donor we can take it. That will help us. Next is after fertilization, the dicot undergo rapid cell division as it travels through the fallopian tube. What is cleavage? It is a type of mitosis where rapid cell division occurs without the interphase or the growth phase. How does it differ from the ordinary mitosis? You know that mitosis will have karyokinesis and the interphase and that cycle. Does the embryo grow in the size during cleavage? No, it doesn't grow in the size during the cleavage. Trace the development stages: zygote, two cells, morula, blastula. How many days does each stage take, and where is the reproductive tract in the embryo to each stage? So you have a diagram in your NCRT, so that would be there. This whole thing from zygote to blastula takes 7 days. Okay, this would be your answer, and if you can make the diagram of that divisions things. Okay, the blastocyst has two distinct cell population. Name them and their position in the blastocyst and the fate of each in the development. So your blastocysts will have what? It will be something like this. It will be covered by the layer, and it has what? It has cells. Okay, this layer is called as tropoblast. This layer is inner cell mass, and this is what? Blastocele. Now these are the what? Two distinct types of cell layer we have made it. Their position also we have showed it, that there will be what? This would be what? This would be embryonal pole, and this will be what? An embryonal pole. Embryonal pole. Their fate: the tropoblast will change into the chorion, chorionic villi, or the placental part. Inner cell mass will form what? Embryo. Not just the embryo, there are other things also will form, but that's the point over here. Okay, next is implantation is a crucial critical event in the established pregnancy. What is the difference between tropoblast and the inner cell mass in the blastocyst? You know that. Which give rise to the embryo proper and which give rise to the placenta? You know that. We have already done it. Next, for implantation to succeed, 3 conditions must be met simultaneously. State the 3 conditions. Nothing, that's not at your level. How does the tropoblast invades the endometrium? What enzyme does it uses? Why must this invasion process be tightly regulated? Very important, this is not in your syllabus. You just have to remember that hatching of blastula take place. Hatching of blastula takes place, and the tropoblast cells which are present, which will go and joins with endometrium, as they have sticky consistency or sticky material which gets sticked with it. Now the next is a woman's blastocyst forms normally but fails to implant. Suggest the 3 possible reasons. 1 reason to the blastocyst, 1 reason to endometrium, 1 reason to the hormonal balance. Actually, these are 3 ways of implantation. But for you, just remember that they can ask that blastula might not be able to stick because of the tropoblast cells issue. Okay, coming up to the next one. These are all the events that would be taking place. The one that you have to remember is this one. On implantation, HCG is secreted. What produces HCG? Chorion, tropoblast. Okay, what does HCG do? It stimulates the corpus luteum to keep on secreting progesterone and estrogen for the next 3 months. Why is HCG the basic for the pregnancy test? Because if HCG will be present after the 7, once the implantation, the HCG amount will only and only be present during the pregnancy only. So it tests that whether you are pregnant or not, because HCG is a pregnancy hormone. At what earliest point of the fertilization can be 7th day? Okay, moving on to the next one, that is pregnancy and embryonic development. The placenta is described as both the mechanical and the biochemical interference between the mother and the fetus. From which 2 tissues does the placenta develop? One is the chorion, one is the endometrium. Okay, what is its primary structural unit? Chorions are its primary structural unit. List 4 hormones produced by placenta and specify the function of it. Human placentogenic hormone, lactogenic human plus lactogenic hormones. All of these things will be secreted. SCG, human chorionic gonadotropin. These things will be secreted by it, and its function you know that from the NCRT itself. Then the placenta allows exchange of nutrients and gases between the fetal and the maternal blood, but the two blood supplies do not mixes. Explain the structural basis, because there is a presence of umbilical cord which contains the umbilical artery and the umbilical vein. Okay, so that thing is there. Next question. Embryonic development involves 3 primary germ layers whose formation is called gastrulation. Name the 3 germ layers formed during gastrulation: ectoderm, mesoderm, and endoderm. From which layer does each of these following? Okay, these things are what? Not at your level. So you can leave this. Okay, this question from here. The inner cell mass of the blastocell is first from the bilaminar disc. Describe what happens during gastrulation to convert it into trilaminar. No need. Okay, this is not at your NCRT level. What is no need? Okay, next comes human pregnancy lasts approximately 9 months divided into 3 trimesters. Describe the major development milestone in each trimester. This is directly the question from your NCRT paragraphs. Okay, then the fetal heart begins beating approximately 4 weeks in 1 month, but the mother cannot feel fetal movement until 4-5 weeks because their limbs will be formed that time. Okay, explain the development reason for this delay. Okay, SCG levels peak at 8th and 10th week of the pregnancy and then decline. What does this peak tells it about the corpus luteum functions during early pregnancy? Why? During early pregnancy, it is regressing. So corpus luteum has to keep on working if the SCG levels would be super high. So it is correlated. If more SCG will give a positive feedback to the corpus luteum to keep on working it and forms the hormone of the pregnancy. Analyze the role of the hormone in the pregnancy. Data: blood level of SCG, estrogen, progesterone during pregnancy. SCG peak at the week 10, then declines to the low level for the rest of the pregnancy. Progesterone rises continuously throughout the pregnancy. Estrogen gradually rises throughout the pregnancy. What does SCG peak at 10? Why does the SCG and then declines? Because at the 10th week, it has to stimulate the corpus luteum enough so that it's keep on producing progesterone and estrogen so that it can sustain the endometrium lining. Okay, progesterone is called the hormone of pregnancy. List the 3 specific effects of the progesterone that maintains the pregnancy. Endometrium lining, the main important thing. Okay, that's it. Those questions are easier and all. Okay, then these are the basic questions that has been asked. Certain questions are beyond the levels which you can remove it, that is NEAT level, like this thalidomide medicines they are talking about. And the last topic is parturition and lactation. This is the easiest topic, okay, if you know the whole thing. So the first question is, childbirth is initiated by the complex hormone and the mechanical cascade. What is the role of oxytocin in the parturition? It contracts the uterine lining and pushes the hormone. Which hormone pushes the fetus? Which gland releases it? It's the posterior lobe of pituitary. Explain the positive feedback mechanism of the parturition. How does this initiate contraction of the uterine stimulates leads to the stronger and the stronger contraction until delivery? So first what happened? Fetus gives stimulation to hypothalamus of female, to the mother hypothalamus. Then mother hypothalamus after stimulation get what? Gets the anterior lobe of pituitary and it secrete what? It secrete oxytocin. Okay, this oxytocin will do what? It acts on the muscles of uterus, smooth muscles of the uterus, and it will do what? Contractions. Now these contractions will do what? Deliver the baby or push the baby outward. Now as the contraction occurs, this gives what? A positive feedback. So more contractions will keep on coming up. Okay, so fetal ejection reflex is there. Fetal ejection reflex is said to be the signal that initiates the labor. What causes this reflex? Fetus causes this reflex. What does the fetus release that contributes to the triggering? That you don't have to do it. Okay. Next is the hormone relaxin plays an important role in the parturition. What it does, it relaxes the pubic symphysis, dilates it, as well as dilates the cervical mucus and the structures through which the canal will be formed. Okay, this more things are being given to you, prostaglandin and all. These functions are not there in your syllabus. Next is the lactation. You can see this question, easy straightforward question, straightforward thing like that. Only two hormone, prolactin and oxytocin, will help in this process, and what they will do, who will secrete it, who will produce it. And there is a one question on lactational amenorrhea is also given to you, which you can also do it. So this is an important question, but very straightforward question. Take a screenshot and try to do it yourself. Okay, if you didn't get it, let me know in the comment section. Coming up to the next one, there is a proper event that has been given up to you. You can just go through this question, and then there is a basic questions based on that, but this is the high level questions over here given to you that you can leave it. Okay, and that's pretty much all about human reproduction. We try to do a lot of questions. I showed you a lot of questions to make your temperament towards the examination, so that when the tough question comes, you can remember these points. If you are able to understand, if you need something more peculiar and more than this, let me know in the comment section. Well, that's all for this video. I will see you in another video super soon. Okay, bye.